So we're going to start in a moment with Dr. Octavio Choi. I saved my sort of thank you now for him. Tavi is also the reason that we're having this program. He is, as you'll read in your bio about him in the briefing book, just one of the premier up-and-coming psychiatrist and neuroscientist, trained both as an MD and a PhD. Tavi is one of a new wave of doctors who are, you want to change your slides? Who are introducing neuroscience into their clinical practice as well. Tavi directs forensic psychiatry here in Oregon, works intensely with the criminal justice system, and is going to get us kicked off with the really basic question of how does the brain work? He has the toughest job of the entire day. I told you tens of thousands of people are trying to figure out. We're giving him about 40 minutes to get us up to speed. But if there's one person who can do it, it's Tavi. And with that, let me hand it off to you.
Thanks, Francis. Okay, microphone's working. This is actually probably the toughest talk I've ever had to put together, trying to explain the brain in 40 minutes. And so let's get to it. So I'm an assistant professor at OHSU, and I am the director of the Forensic Evaluation Service at the Oregon State Hospital. In that capacity, I have the privilege of living neurolaw, boots on the ground, evaluating criminal defendants with various types of medical and brain conditions, and assessing how responsible they might be for the offenses. So let's start with a case that something that you might all be in a position of considering. Imagine there's a defendant who was causing a public disturbance, was urinating in public, swearing in public, and this is a 25-year-old man, no criminal history, excellent physical health, never missed a day of work in his life, alert, oriented, aware, intelligent, using language very well, good memory, intact memory, could learn new things. And basically, when you ask him about why he did it, he just says, well, I didn't care. I just didn't care. Now, imagine if 2 months prior to this incident, a tamping iron, 13 pounds, 43 inches long, inch and a quarter in diameter, shot through his skull and penetrated the front of his brain. Would that kind of affect the way you might think about how guilty this person might be for their acts? Might that, in fact, be a strong reason for why this person behaved the way they did? A lot of you are smiling and nodding your heads. Many of you know this is the famous case of Phineas Gage, the most famous patient in neuroscience. I think half of cognitive neuroscience textbooks start with the story of Phineas Gage. So who is Phineas Gage? He was a railroad foreman. Back in 1848, suffered a terrible accident, and from the Boston Post, he reported it. So foreman on a railroad in Cavendish was tamping down, he was exploding rocks to make way for a railroad track, and you have to put, gunpowder or the explosive, and you tamp it down with a tamping iron, which caused a spark in the rock, and it exploded, and the tamping iron was driven through his skull. 100 years later, back in 2004, Phineas Gage's skull is currently at the Harvard Museum, and the doctors got his skull, imaged it in a CAT scanner, created a 3D model of the skull, and was able to digitally remaster exactly the path of the tamping iron. So this is just showing off that we have the skull. We're kind of creating a bunch of slices with this CAT scan, and reconstructing it with a computer to make a three-dimensional model that's completely accurate in every detail. This is part of the computer revolution. With increase in computing power, you can take these two-dimensional slices and make three-dimensional models. And there you see the tamping iron going through a skull. Ouch. Now, one of the amazing things about Phineas Gage is that he survived. And in fact, he was treated by a physician named John Martin Harlow, who became famous by reporting on the case of Phineas Gage. And here's what he had to say. I'm going to just read this because he's such a good writer. The equilibrium or balance, so to speak, between his intellectual faculties and animal propensities seems to have been destroyed. He is fitful, irreverent, indulging at times in the grossest profanity, which was not previously his custom, manifesting but little deference for his fellows, impatient of restraint or advice when it conflicts with his desires, at times pertinaciously obstinate. I love that. Yet capricious and vacillating, devising many plans of future operations which are no sooner arranged than they are abandoned, in turn for others appearing more feasible. Sound like anyone we know? I mean, a lot of us who work through, with the criminal defendants, I mean, this describes a lot, kind of very impulsive kind of personalities. But the difference with Phineas Gage is that before his injury, although untrained in the schools, he possessed a well-balanced mind, was looked upon by those who knew him as shrewd, smart businessman, very energetic and persistent in executing his plans of operation. In this regard, his mind was radically changed, so decidedly that his friends and acquaintances said he was no longer Gage. Now, Phineas Gage is often pointed to as sort of the start of the birth of modern cognitive neuroscience. And why is this? He was really, for the first clear example written in the literature of someone who suffered a brain injury and had very specific impairments, which pointed to the key principle of the brain, that different parts of the brain do different things. So if you damage different parts of the brain, there will be specific impairments that result. So the key phrase is the equilibrium or balance between his intellectual faculties and animal propensities seems to have been destroyed. So his intelligence, as it was thought of back then, was intact. His memory was intact. He was able to have a conversation with you. He knew where he was. He could learn new things. All that stuff was intact. And also his emotions were intact. He was able to feel happy and sad and all those things. But the balance between those two things was destroyed. And I would argue that the best way to summarize Phineas Gage's impairments, his brain deficit, was that his conscience was impaired by that specific brain injury. So what is a conscience? Anyone just want to throw out? His conscience was impaired, in my opinion, just my opinion. But so what do we mean by conscience? Jiminy Cricket. And what about Jiminy Cricket?
The voice on your shoulder telling you this is the right thing to do, that's not right.
Okay, so it kind of helps you in the right between the wrong. And is it Jiminy Cricket where it's a cognitive thing, where it's like you know the rules are wrong, or is it like an emotional thing that stops you from doing what you think is right or wrong? It could be both, but what do you think is more important? Yeah. Anyone have this thing where you're in the middle of a kind of a deserted town in the middle of the night, and you're stopped at a traffic light at the red light? There's no cops around, right? But what do most of us do? We're glued to that to that red traffic light, right? We're not going forward, even though we know there's no chance we're going to get caught. So what is it that's stopping us? The limbic brain. Okay, but explain it for people who don't know what the limbic brain is.
It's that gut consciousness that you can't, it's feeling, and it has no cognitive skill building, it's just a feeling.
Yeah, I would argue that it's sort of like this bad feeling in your body that you get automatically when confronted with these kinds of dilemmas. And it's that bad feeling, I would argue, might be more important than the rational, I know this is wrong, kind of, cognitive thinking. So, the weird thing, or the interesting thing about Phineas Gage is he seems to have suffered an injury that impairs that part of the brain which allows you to call up bad feelings in response to something that you know is wrong. So we'll get back to that in a moment, but let's just go over some basic neuroanatomy. So this is the picture of the brain that many of us are familiar with. This is the cerebral cortex. You can see this is tough with a split screen. I only have one pointer, so. This is the cerebral cortex. We can see that this is the surface of the brain. We can see it split into various lobes. There's the frontal lobe in the front. There's a temporal lobe next to the temples. The back of the brain is called the occipital lobe, and the parietal lobe is in between. And here is a top-down view. So we're looking from top down. You can see the eyeball. So it's like the brain is pointed this way. And you can see, an odd thing about the brain is that, like the right side of your brain controls the left half of your body, and the left side of your brain controls the right side of your body. So you guys probably know people who've suffered strokes, and the interesting thing is that the site of the injury often results in deficits on the opposite side of the body. These two halves of the brain are connected by a bridge called the corpus callosum. It's a huge a series of connections that connect the right and left halves of your brain. And in the coffee break, asked me about split brain experiments. And neurosurgeons have actually cut this bridge. And it turns out what happens is that splits you into two different people that live in the same body. So it's a really a fascinating thing that we're not going to go into more in this talk, but there's a big bridge called the corpus callosum. And then this is a picture of the brain looking as if the brain is looking right at you. And so you can see the corpus callosum is this bridge. And you can see that there's stuff buried underneath the cortex, right? So that's a really important point. So the brain is not just what you see on the surface. There's stuff buried underneath there. Oh, another thing is you can see that the brain has lots of convolutions, and the bumps are called gyri, and the dips are called sulci. And why do you think we have all these convolutions and dips and sulci and stuff like that? What does that do? Increase the surface area, right. So if the brain is a computer, you can pack more computing machinery into that same area. And actually, if you look evolutionarily, these are different kinds of primates in the relative sizes. So here's humans, and we have the biggest brains, but we also have the wrinkliest brains. So when you look back in evolutionary history, the brains get smoother and smoother. So it's a very interesting thing that we get more brain machinery stuffed in there by all those folds. So another key point is that the brain is a multi-layered structure. It's not just the cortex, what you see on the surface. There's lots of machinery underneath there. So a lot of us know about this theory called the triune brain, which was put forth in the 70s by a Harvard psychiatrist named McLean. Basically, he posited that when you look at the modern human brain, you can conceptualize it as being constructed of 3 different layers. What he called the reptilian layer, the limbic system, and then the neomammalian, which is cerebral cortex. So if you look at a MRI, and here's the nose looking forward, here's the brain, we consider this first most primitive part of the brain to be called the reptilian complex, which is the brainstem. On top of that is built a series of structures called the limbic system, and on top of that we have the covering called the cortex. Cortex literally means covering. So here's the cortex, and what the cortex in general does is it manages all the computations required for higher cognition. Stuff like language, stuff like higher order object recognition, telling the difference between a Van Gogh and a Rembrandt. Consciousness, being consciously aware of your thoughts, consciously aware of your feelings. All that fancy stuff that we consider being human tends to be computed and instantiated in the cerebral cortex. If we get rid of that upper layer, we see underneath it a very complex series of structures that I'm just going to simplify and call the limbic system. For the hardcore neuroscientists, I know that that's kind of a simplification, but let's just call this middle layer the limbic system. And what this and the limbic system does many things, but one of the key things it does is core evaluation. So, our brains evolved to keep us alive. And what the limbic system does with core evaluations is it scans everything on the outside, hearing, sight, sounds, touches, feelings inside our body, and it just scans for anything that you might want to pay attention to because it's going to help you survive. So things like donuts, things like spiders, things like sexual partners, right? So if it sees, it's constantly scanning. If it sees something that's going to help you survive in some way, it goes ding-a-ling-a-ling-a-ling and tells the cerebral cortex. So then you can actually go towards that donut, away from the spider, away from the, right? And it doesn't, that message, what does that message feel like? It doesn't feel like a text message coming up on your phone. It feels like the feeling of craving, like the feeling if you want to go towards something, or a feeling of terror or fear to move away. So these core evaluations are computations done at a very low level that scans the entire external and internal environment in order to give your your cortex a signal to move towards things or move away from things. And those signals feel like feelings. If we get rid of the limbic system, underneath that we have the brainstem, which has a bunch of basic life support. So the brainstem is actually the upper extension of the spinal cord. Can you guys see okay here? I feel bad because my pointer doesn't show up on this thing, so I'm sorry. Maybe we'll do like a Bono. I went to a U2 concert, and it was a circular stage, so half the concert he was this way, and the other half he was this way. So maybe midway I'll switch over. So the brainstem is the upper extension of the spinal cord, and it handles a lot of basic life support. There are centers there that control respiration, heart rate, and basic sensory processing. There are sensors there that are constantly sampling the chemical environment of your blood to make sure there's enough oxygen in it, for instance. So lots of basic life supports in there. If you have a stroke in your brainstem, you generally die. Like, you don't have breathing, you die. Okay, so there's the brainstem. On top of that, we have the limbic system that is kind of constantly looking out for things at a low level to help you survive, core evaluations. And on top of that, we have this thinking brain, the cortex. Now, another key principle is the brain is hierarchically organized. So, you are not aware of most of the things your brain is doing, right? Anyone here consciously beating their heart? Or, can you forget how to breathe? Not really, right? That's because your brain, the lower centers, the brainstem, the limbic system, they're doing lots of things underneath consciousness. They're helping you stay alive. And your cortex is only notified when, it's just like a corporation. You have your underlings do most of the grunt work, answering the low-level emails. And anytime there's something, I think the boss needs to know about this because there's two conflicting emails and I'm not quite sure how to resolve it, that then gets bumped up to the next level of the hierarchy. So for instance, your brainstem has basic life support systems, including sensors for oxygen levels in your blood. So if there's a low level of oxygen if the oxygen level in this room drops down, these sensors are going to start getting very excited, saying, there's something wrong here, I need to notify the boss. The brainstem's boss is the limbic system. The limbic system it's monitoring everything, even stuff outside of your awareness, and that's really cool. Like you're not aware of, the feeling of, the air on the back of your left hand, right? I mean, now you are, but you weren't before. But I guarantee you, if there is a feeling of some fuzzy legs walking on the arm, suddenly that limbic system would say, this is important, I need to tell the boss, and you become aware of it, okay? So if you have drop in O2, that tells the limbic system. Limbic system's sampling lots of things, including the feeling in the back of your hand. But if it gets a message saying your O2 level is dropping, it says, I need to tell the boss about this. This just can't go on, right? And so then it sends a signal to the cortex. Cortex says, yes, I do feel like I'm suffocating. And so I'm going to get out of here and possibly go outside. But please stay in the room. So this is a busy diagram. This is a diagram from one of the references that we put in the briefing book, one of the latest issues, articles about the brain circuitry of drug addiction. And so, don't get overwhelmed. It's easy to get overwhelmed with these kinds of diagrams. I just want you to pay attention to three different areas: the amygdala, the ventral striatum, and the orbital frontal cortex. Right, so I have to give some love to this side. The amygdala, the ventral striatum, and the orbital frontal cortex. Now, the amygdala and the ventral striatum are part of the limbic system, doing those core evaluations. The amygdala tends to be scanning for things that could possibly be threatening to you and you need to move away from. And when the amygdala activates, that feels like fear, get away. The ventral striatum is colloquially known as the pleasure center of the brain, and these are all oversimplifications. Every brain area kind of does everything, but just to keep things a bit more simple. The ventral striatum is called the pleasure area of the brain, and it tends to be looking out for things like donuts, sexual partners. It gets excited when it sees things that you might be interested in moving towards, and that feels like pleasure, feels like anticipation. If it activates and the thing isn't there, like if you want a donut but there's no donut there, that activation feels like craving. Now, obviously, you don't want to act on every single impulse your amygdala and ventral striatum is saying. You don't want to, unless you're a presidential candidate, if you see an attractive sexual partner, you're gonna generally want to say, I couldn't resist. I'm sorry. But that's where your cortex comes in. So your orbital frontal cortex is involved in evaluating the signals coming from the amygdala and the ventral striatum, saying it adds context to what those drives are telling you to do. It says, well, maybe we shouldn't grope that person, or maybe we shouldn't have that sixth donut, or the amygdala is like the amygdala is like a scared little kid that everything freaks that little kid's out, So it sees a spider and the amygdala goes, oh my God, there's a spider. It gets very active and excited. And the orbital frontal cortex adds context. It says, you know what? There's a cage around that spider. We're actually in a zoo, so calm down, amygdala. It's going to be fine. Or the ventral striatum gets very excited, seeing a donut, and the orbital frontal cortex says, the last couple times you kind of overdid it with the donuts, and you felt really bad. So it kind of helps modulate and inhibit those drives that come from underneath in the limbic system. Well, guess what part of the brain was damaged in Phineas Gage? The orbital frontal cortex. The tamping rod went straight through that area. That's the area that gives emotional context to the drives and can inhibit appropriately the drives that should be inhibited. And so Antonio DiMaggio and his wife Hannah, Antonio DiMaggio is one of the geniuses of neuroscience. He's a neurologist. He used to be in Iowa. I think he's moved around. He's at USC now. Some great folks at USC. And he studied this area of the brain called so it's the orbital frontal cortex. Here's Phineas Gage's tamping iron. It went through, and here's a side view of the brain with the brain looking forward this way. and then underneath the ventral surface of the brain, the eyeballs would be here. So you're looking underneath. This section of the brain is called the orbital frontal cortex. It's also called the ventromedial prefrontal cortex. That was the area that was damaged in Phineas Gage. And his lab was really instrumental in studying stroke patients to understand what this part of the brain was doing. Because the fascinating thing is, if you damage this part of the brain, your memory is fine. You know where you are. Your use of language is fine. Your cognition is fine. And also your emotions are fine. They're able to feel happy and sad. But they no longer seem to care about inhibiting things that we would find socially kind of repulsive. Right? So I would argue that that's what keeps us from urinating in public, even though we have a really full bladder. We're just not going to do it. The reason we don't do it, yeah, we know it's wrong, but the other reason is we know we would die of embarrassment, right? And that feeling, it's a feeling that stops us. It's this feeling of, I feel really bad in my body contemplating doing this. And that's what generally stops us from doing things. And where did that feeling come from? How do we get that feeling in our brain? Any of you have babies? Do they have problems urinating in public? Absolutely not. We learned that, right? It's a series of learned emotional response. It's like you did something, you urinated in public, and then you felt bad somehow. Mommy said, don't do that, and you felt bad in your body. Over time, you developed an emotional linkage of a state of your body with a certain context. That is exactly what this part of this brain is doing. So people with damage in this part of the brain, they have something called emotional amnesia. They can feel emotions, but they just don't store it, so they can't form those linkages between a context and a feeling in their body that would ordinarily stop many of us from doing stuff like urinating in public or groping people. Okay. So DiMaggio, the way he put it is this area of the brain holds linkages between the facts that compose a situation and the emotion previously paired with it. And I would say that is actually a major part of what the conscious is. This is one of the main brain areas where the conscience is instantiated. Okay, and in general, Phineas Gage was an important case of someone who suffered a specific brain injury and had a specific deficit, thereby showing a key principle that different parts of the brain do different things. That principle is called functional specialization. And actually, the man sitting there, Michael Posner, is one of the key people who established that principle in the brain. And so, this idea that different parts of the brain do different things, that might sound like a really obvious idea to modern day people, but it was not an obvious idea 100 years ago. And in fact, there was a raging debate between the holists and the localists. So Pierre Florenz was one of the main proponents of holism, which said, like, the brain is a computer and all parts of it are undifferentiated. So really, impairments don't have to do with the location of the impairment, it has to do with the size of the impairment, of the damage. Right, so every brain part is kind of interchangeable. Franz Joseph Gall was a proponent of localism that said, no, brain areas are highly specialized. Okay, different parts of the brain do different things. But he took it a little too far. He's the main guy for the theory of phrenology, which you guys know phrenology, right? Bumps on the surface of your head means you're a criminal, it means you're a good person. I have a very bumpy head. I don't know what that. I shaved my head to go on a meditation retreat a few years ago, and I was like, oh my God, I'm never shaving my. That's why my hair is kind of like, God would hate me. But he took it too far. He said, the brain is divided into areas, and there's an area for acquisitiveness, and there's an area for whatever sublimity is, and an area for mirthfulness. And there's one area of the brain that does that, and that's nonsense. That's taking it too far. Because when you think about it, any task that you do, like let's say you're reading, right? When you think about what that task involves, it involves looking at words, so there's a visual system involved in it, right? Then it's understanding the words, so there's processing of what that visual signal means. It might invoke memories, right? So, and those memories might invoke kind of behaviors or decisions you might want to make. Each of those different qualities of that task is done in different parts of the brain. So even something as simple as reading a single word activates different parts of the brain associated with those elementary tasks. Does that make sense? And that's what Dr. Posner's work with his colleagues at Washington University established in a series of super elegant studies. I love reading those studies. When I read those studies, I feel like, oh, I understand the brain. Okay, so actually, localism's taking it too far. Holism is also taking it too far. There's this in-between thing called distributed processing. And that's the principle that I just elucidated where, it's not that there's one part of the brain that does, a single thing, but any task that you have has elementary cognitive operations, and each of those elementary operations are done by a different part of the brain. Does that make sense? So it's a collection of specialized modules that gets invoked dynamically depending on the task that's at hand. Right? Okay, good. Nodding heads. Good. Now, Phineas Gage was a loss of function study. Tamping iron went through a certain part of his brain, and he lost some ability. And so people started to make correlations between parts of the brain and the parts and what kind of functions that are done by that part of the brain. Many of us are probably familiar with stroke patients. So that's another loss of function study where part of your brain gets knocked out. And by studying what kind of impairments result from that, you can make inferences about what that brain area is useful for. Right? Make sense? In fact, some of the seminal studies, early studies on brain localization had to do with aphasias or language deficits. And so you guys heard of Broca and Wernicke? These are two famous scientists, neurologists around the turn of the century who studied some fascinating patients who had language deficits in different parts of their brain. Let me go over here for a little bit. So Pierre Broca was a French neurologist who found that a bunch of his patients lost the ability to vocalize language. So they could understand language when you spoke to them, and they could execute commands. If you said them for them to do something, indicating that they understood language. But they had a really hard time generating language and speaking. So he discovered that these patients had a damage in an area of the cortex called Broca's area. And interestingly, that Broca's area is right next to the part of the brain that controls the motor the muscles of the lips and tongue. So damage to this area results in impairments where patients can understand language, but they have a hard time generating language.
Boy is that landing down.
It's hard to hear, but
is
this person's having a whole lot of
laughing,
time expressing language.
And cookie jar.
You would call this person fluent, right? She has damage in Broca's area. Now, there's another area called Wernicke's area, which is close to the auditory cortex, where with the part of the brain that processes incoming sound signals. Damage to the Wernicke's area results in a language deficit in which people can generate language just fine. They're talking up a storm, but they can't understand language, spoken language. They can understand written language, which comes in through a different pathway, through the visual cortex, but they can't understand spoken language. So when you ask them to do something in a verbal command, they respond with very fluent nonsense. So here's an example.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck, for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here and they'll save their hands right there for them.
And what
Okay, so that's Broca's area. So those early studies were super interesting and important for establishing, again, that different parts of the brain do different things. Also, around the turn of the century, a guy named Broadman had a microscope, a novel technology at the time. By sampling little core samples from different areas of the cortex and looking at them under the microscope, he could see that cells look different in different parts of the brain. And he hypothesized that the reason those cells look different is because these brain areas are specialized and do different things. So this is a Broadman map, and many of you might have seen this. We still use this map of brain areas, which he numbered, like area 17 is primary visual cortex, and neuroscientists still refer to Broca's areas. So it's another indication that if you look at the microscopic level of different parts of the brain, it looks different, and that supports the theory that different parts of the brain do different things. Any questions so far? Or, yeah, this is all good? Okay. So now Broca, I learned this thing, trigger alert. Trigger alert? I hadn't heard this before yesterday. I need to warn you for anything that might potentially be traumatic. And my tone of voice was a little sarcastic. I don't mean that. But Wernicke and Broca, they discovered where the damage was in their patient's brain they had to wait until those patients died. And then they had to slice up those brains, literally with a knife, and then find little goobers and stuff. And it's like, oh, okay, this seems to be where the problem is. Now you can imagine that's an incredibly slow process, right? Like you have 5 really interesting patients, and you have to wait 10 or 20 years until they die, or unethically hope that they die sooner. Now, wouldn't the whole enterprise be a lot more efficient if you could look to see where the damage was in the living person, right? That would be fantastic, right? And so that's what the imaging revolution has done to help us. So I'm going to talk about sort of the evolution of brain imaging. So the X-ray, we all know about X-rays, invented or discovered by Röntgen. He reported in a meeting in 1895 that he discovered these X-rays, and this is the very first X-ray he took of his wife's hand, and you can see the ring. And his wife was reputed to have said, "I have seen my death," when she saw this scan. Now, X-rays are amazing. X-rays work by shooting high-intensity waves called X-rays through a person's body, and on the other side is a photographic plate. And then some things stop X-rays more than others, like bones stop X-rays really well, leading to light areas in the X-ray, and soft tissues tend to stop them not so well. They go through, and so the photographic plate gets dark. So they're called shadow pictures. And here's a skull X-ray, and the dream was maybe we can see the brain with this X-ray stuff, right? But the problem was the brain is inside this thing called the skull, and the skull is a really dense thing that stops most X-rays, and so it's really hard to see. Also, with an X-ray, a 3-dimensional thing is flattened into 2 dimensions, so it becomes really hard to tell anything apart. That's why radiologists get paid the big bucks. They see something like this, and they can turn that into a 3-dimensional image in their heads. So for a long time, the brain was called the dark continent. We had no way of imaging it. Now, I think around the 50s, 60s, someone had the bright idea of injecting air into the brain, because air is very not dense, and compared to brain tissue, we might be able to tell the difference in the X-ray. So the brain has a series of interconnected fluid canals called the ventricles. Right? So the ventricles make cerebrospinal fluid and bathe the brain in fluid. Someone had the bright idea of taking a big syringe of air, injecting it through the brain, and injecting air into the system, and then you'd be able to see the bubble of air with an X-ray. Later on, people said, we actually don't need to go right through the brain. We can go through the spinal cord with a spinal tap kind of procedure, and that was a lot better. But still, these are the kind of images you get, right? It's going to be hard to tell. You can tell if someone has a major problem with their ventricles, you can tell something's going on. But it's a low-resolution image. And I like seeing this to remind us how far we've come. You inject the air bubble, and then they're in this chair that kind of puts you in different positions. And then you take X-rays in those different positions as the air bubble moves to kind of. This was supposed to be an incredibly painful procedure, and the joke was that this procedure was therapeutic because the patient, whatever their complaint was, never came back to you again. So this was the 60s. Okay, shooting ahead, the CAT scan. It was invented in the 60s. The Beatles were an incredibly important part of developing the CAT scan. The CAT scan was developed by EMI, which is also the record label of the Beatles. EMI ended up with just a ton of money from the Beatles albums. They said, what can we do with all this money? An engineer at EMI said, I know what to do with that money. With the rise of computing power, I can develop a new kind of X-ray that will take a series of X-ray images in a rotational manner, and with my computer algorithms, I can reconstruct a 2-dimensional and then a 3-dimensional structure of the brain. Amazingly, it worked. So when you look at a CAT scanner, this is the inside. What this is a spinning X-ray, where you have an X-ray source that's spinning in a circle, and there's a photographic plate, and it's taking like 1 million X-rays, right? And then magically, if you do take 1 million different X-rays from different angles, reconstruct that with a computer, you can now start to look at brain tissue. So this is the first CAT scan. And can you say anything here? There's a dark spot right there. So it's fantastic. It's always great when the first CAT scan shows something you can actually do something about. So this turned out to be a tumor that was resected. And Hounsfield went on to win the Nobel Prize, and it was all good. The MRI is a technology that most of us are familiar with. It's our modern imaging method, and it's a much bigger machine. So here's a person, and the MRI, and it'll take hours of physics to explain exactly how it works. But suffice it to say that the MRI can interrogate magnetic signals all throughout your brain, and because your brain is made of different compositions of water and fat and different kinds of molecules, it can actually distinguish those magnetically. So those have different magnetic properties that can be converted to an image. And the MRI resolution is dependent on magnetic field strength. So the early MRIs were 0.35 Tesla, then there were 1.5 Tesla, then you went up to 3.0 Tesla, then you went up to the modern research scanners have 7 Teslas. And if you compare the difference between 0.35 Tesla and 7 Tesla, there was a remarkable improvement in resolution. The world's most powerful MRI is being constructed. That's a person inside there. It's going to generate an almost 12 Tesla signal, which is just enormous. And with that enormous magnetic strength, the voxels, or the brain elements that get imaged, get smaller and smaller. So we're going to get incredibly high resolution pictures of the brain. And like many big science projects, it keeps getting pushed back. Now, that's loss of function studies, which are really useful, but it's sort of akin of, like, if you want to figure out how your iPhone worked, it's like if you took an ice pick and stabbed different parts of your iPhone and said, oh, well, this doesn't seem to work anymore, right? It's kind of a slow, laborious process. So what we'd really like to do is study the intact functioning brain. What is the normal structure and function of the brain? And so that's where functional imaging studies come in, and that's really a revolution. So the two main ways to visualize brain activity these days are with PET and with FMRI. PET studies are amazing, and they can do things that fMRI can't do. I'm going to focus mostly on fMRI, which is used more. PET requires injection of radioactive particles, and a lot of patients kind of don't like that so much. So fMRI, it's all done with magnets, so it's safe, and it's even holistically beneficial. So fMRI. So the thing with fMRI, a key point is that, although these pictures are very pretty and it looks like we're looking at brain activity, we are not looking at brain activity. We are looking at an indirect measure of brain activity. We're actually looking at a vascular response to brain activity. So the fMRI visualizes a vascular response to brain activity, not brain activity itself. And so, the basic principle is if a part of your brain is working hard, it's going to recruit more blood flow. That extra blood flow is going to change the magnetic properties of that part of the brain, and that can be visualized on the fMRI. So the fMRI actually visualizes what's called a relative oxygenation level, which is an indirect measure of brain activity, because your brain's working hard, recruits more blood, oxygen level goes up, and that can be seen on the MRI. Now, so this is an important slide, and I see the references in the briefing book. There's many limitations of fMRI. We're looking at a correlation of vascular response to brain activity, and all correlations are imperfect, and there's people who've made their careers studying how those things can be mismatched. The other thing to really understand is that this is a highly processed image. This is not just a photograph of your brain activity. This is a highly refined image with many steps and statistical assumptions behind it. There's a lack of standardization, so that one lab's fMRI machine has a bunch of settings that aren't the same as another lab's fMRI machine. So you can imagine if this gets into court, and it's really been resisted in courts because one lab's fMRI image can look very different from another one's if all the settings are different, right? There hasn't been standardization in the same way that DNA analysis has been completely standardized from lab to lab. And also, some of the statistical assumptions have been shown to be faulty. In fact, there was a major paper this year that was incredibly disturbing to fMRI researchers that showed that based on an obscure setting in the software of most fMRI software packages, that up to 70% of the results might just be BS. So it's still a new technology. It's developed in the 90s, but it's still relatively new. fMRI is being used more and more. And in fact, Sarah Felton Ewing will present, I hope, on her elegant studies showing, or I'm not going to tell her what she's going to present, but she's done very elegant work looking at adolescents engaging in therapy, seeing what parts of their brain are active, and using those fMRI images to refine what kind of therapeutic interventions might be the most effective. So different parts of the brain do different things. That's how we get these colorful maps that you can see if you open up any web page. Another key principle, brain areas are wired together, right? They do these different things, and they talk to each other. They form these local circuits. Right, so this is a circuit that we've talked about already. There's the amygdala, the part of the limbic system that gets really excited over things that are scary. The ventral striatum, the part of the brain that gets excited over things that like donuts. And the orbital frontal cortex, it says, hey, maybe we shouldn't follow every impulse that we might have. And in fact, what Dr. Compton, I think, will talk about is how this circuit gets corrupted as you develop a drug addiction. when you look at the microscopic level of what these wires are composed of, they're composed of brain cells. So here's a high-resolution picture of a brain cell with a cell body, the wire, which is called the axon, and then the end of it, which is called the axon terminal. And so this is the basic computing unit of the brain. And what's interesting about this is that this is a chemical system. So the dendrite has, the cell body has lots of extensions that sniff around the chemical environment, looking for things that it makes it excited, chemicals that make it excited. If it gets excited, it generates an electrical charge that then gets sent down the axon in an electrical impulse. And at the other end, it releases chemicals called neurotransmitters, which then continues the brain communication. That's why the brains are, you can control brains with electrical, you can put an electrode in someone's brain, turn it on, and see weird things happen. You can also give people drugs, right, medications, different kinds of chemicals, and that will affect brain function. That's because it's affecting the chemical environment which gets picked up by the neuron. Does that make sense? Okay, and this is a high-resolution view of what the end of one axon with the start of a new neuron. So here's a two-neuron circuit. Here's the axon, the wire, right? And then it's connecting to the second neuron, and this connection is called the synapse, right? So I'm going to show you a high-resolution view of the synapse. So the electrical impulse called the action potential comes down. At the end, it releases chemicals called neurotransmitters, in this case acetylcholine. Those neurotransmitters diffuse across the synapse and bind to receptors. So here's a receptor. It binds to the receptor, and what that binding does is open up the pore, which allows electrically charged molecules to come in, which can continue the electrical process. Here's an incredibly impressive example of brain wiring diagrams. Carl Dijksterhoff is a name you should all be aware of. He is 100% guaranteed to win the Nobel Prize. One of the things his lab invented was a method of making the brain clear. So this is a mouse brain, and he made it clear. Once you make it clear, you can fluorescently label brain cells and look at circuitry at a whole brain level. So here's one of the really impressive videos from his lab, where he's showing you the mouse brain. He's showing you how they're taking two-dimensional slices, which they're going to feed into a computer to construct a completely accurate three-dimensional model, high-resolution model. You zoom in, you can see this is the whole brain. These are the axons. You can see axons coursing back and forth. And it's all in the computer, so you can rotate. You can make parts of it transparent. So now the computer's reconstructing the three-dimensional model, which is then you can then rotate. And you can then fly through the brain, look at cellular level resolution. I mean, imagine this 100 years ago. This is an incredible technology. Carl Dijksterhoff. Yeah. This is the part of the brain called hippocampus, which is important for memory. And again, he's just slicing through. These are the cell bodies. You can see these wires are the axons here. He's labeled different sets of neurons with different colors. He can then take each slice, reconstruct it with a computer, make a three-dimensional model. And this is not an artist's rendition. This is an actual part of the brain with all the wiring intact. Okay. In the human beings, we can image this with diffusion tensor imaging. Diffusion tensor imaging allows us to see the wires in the brain. It does that because when you look at neurons, they have water in them, and water tends to go up and down the axon. DTI imaging looks at the movement of water molecules, and from that you can infer where the connections in the brain are. So we have these very high-resolution DTI imaging maps of different. These are all the different kind of wires in the brain, and we get those wires by looking at where molecules of water are going back and forth. Okay, I'm going to just finish up with one last slide. So I talked about the brain. It does core evaluation. The limbic system is looking out for things that help you survive: donuts, sexual partners, spiders, move away from, move towards. Right? And then the upper parts of the brain, the cortex, puts those core evaluation into context. It's cognitive context, saying, well, you ate your sixth donut and you didn't feel so good. Emotional context, where you felt bad in your body when you over-binged on donuts before. So it says maybe we shouldn't listen to the core evaluation so much. And that's built up through past experiences. So if you see a donut, your ventral striatum goes ding-a-ling-a-ling, ding-a-ling-a-ling. So that creates that feeling of drive, of wanting to move towards that donut. And I'm from Portland, so that's why there's so many donuts in my talk. We have fantastic donuts. And that, it brings up facts and reasons. Donuts are good, donuts are tasty, donuts have 8,000 calories. It also brings up emotional context of these linkages of what happened and how you felt the previous times you ate donuts. These are all connected in a circuit, right? And then from the activity of the circuit, you come up with a decision: eat or don't eat. And if there are defects in either brain areas or their connections, you're going to have impaired decision making. And a lot of what happens with drug addiction is that this circuit gets corrupted over time. So you make worse and worse decisions. I just want to make a shout out for, this is the last slide. So, how do we change the way we think about things? Well, generally with psychodynamic therapy. With therapy, kind of reframing what the cognitive meaning of something is, right? How do we work on the emotional context? Well, we have therapies like CBT, which change the kind of emotions we actually link to a certain situation. And Sarah might talk about her work or not, but she's doing fabulous work looking at different brain areas that are activated by different kinds of therapy to guide what are the active ingredients that make therapy effective. Now, you can control the core evaluations with medications and with new techniques that I hope our other speakers will talk about. Marion Fireman is going to talk about medications that might affect the activity of these levels, maybe turning down the ventral striatum activity a little bit so you have less craving. And Virginia Cruzon-Carlson and Kathleen Grant, I hope we'll talk about just some fabulous research using DREADs, which are designer drugs that can activate receptors in just very specific neural circuits to turn them up or down. So imagine if there was a tiny little spot in the ventral striatum that led to craving for cocaine. Imagine there was just one spot there. Imagine if you can selectively turn down the activity of just that one spot and get rid of craving for cocaine. That's the promise of the kinds of approaches that these guys are trying to do. We have a great day ahead of us. Thank you so much for being here, and it's a pleasure to talk to you guys. Thank you.